Welcome everyone. Thanks for coming. Welcome to this year's SCIEX Innovation Showcase. It is a pleasure to be here to deliver this annual snapshot of our innovation journey and to take you through what's new from SCIEX this year, 2023. For me, this is always a chance to reflect on why we do what we do at SCIEX, why we spend every minute of every hour of every day pushing ourselves to deliver groundbreaking innovation, and how that is going to help empower our customers to solve their most impactful analytical challenges. I always try to keep in mind what this means in real terms. Picture some of the leading causes of death globally: heart disease, leukemia, Alzheimer's. Even while we sit in this room right now, thanks in part to SCIEX customers, progress into treatment, even potential cures for these diseases is being made at an ever-increasing rate. That pace, that progress has become something of a new normal for science, a rewriting of the rules, where the demands of the speed at which progress can be made is constantly redefined. At SCIEX, we see this as an ever-changing landscape of science as an extraordinary opportunity, and we believe we are uniquely positioned to do so. One of our customers described working in the biopharma today like being on the launch pad of the space race. Although there's nothing as visible as reusable rockets falling out of the sky and landing safely back on the Earth, what is being done in the fields in which we serve is no less miraculous. There is no doubt that we, and I mean we, are in the midst of a revolution. There are medicines in laboratories right now, various stages of discovery and development, that will treat or even cure disease that just a few short years ago were thought of as completely untreatable. SCIEX's role was to answer this call to action by creating disruptive technologies that can unlock scientific thinking, new platforms that can blaze new paths through the barriers that are holding customers back as they rise to meet this opportunity. A recent quote from one of our customers really sums up why this is important. They said, we do this to do something positive, to take action, to help humanity. We do it for the things that might have a tangible effect on people's lives. And so, it is my pleasure to show you today how our latest innovations will help drive impact, how we'll do this through the words and work of our customers who are, after all, the most important part of this equation, as it's the questions they bring that drive our commitment to bring these new technologies to market. And with that, I'd like to introduce Susan Darling, our Senior Director of Product Management. Susan.
Biopharmaceutical product development scientists want confident and quick decision-making. The faster these drugs can be developed and brought to market, the sooner patients have access to life-changing medicine. In addition, the complexity of biotherapeutics continues to compound as new moieties and creative scientific thinking shape the molecules that are currently being developed. To meet these challenges, our customers need a step change in innovation. Our technology can help unlock ever more ways of working and thinking so biopharma scientists can break through these barriers. As a result, we've invested in the development of a platform that can continue to deliver new innovation in biopharma over time and to support the ambition of our customers, which is to deliver new life-changing therapies faster. With the BioPhase 8800 system, we significantly increased throughput of assays for critical quality attributes by multiplexing 8 capillaries at once. We built on our leadership in capillary electrophoresis and established a new platform that can be further developed to deliver new assays for biotherapeutic characterization over and over again. With the Xenotoph 7600 system, we established a new class of accurate mass systems, a platform for biotherapeutics characterization that gives researchers a deeper level of information through disruptive innovations like EAD fragmentation. In doing so, we have paved the way for new transformative characterization approaches on future iterations of the platform. Working with leading pharmaceutical companies and CDMOs in the field, we have seized the next great opportunity for analytical platform innovation. We heard how the lack of data continuity between CIEF and LCMS approaches created delays in the progression of drug candidates through the pipeline due to unknown peaks in CIEF traces that couldn't or were really challenging to be identified. In some cases, non-viable candidates could be progressed through the pipeline only to fail at a later stage, incurring significant cost. We also heard how process development for novel therapeutics was being hindered by the lack of comprehensive characterization data. I'm extremely proud to announce the launch of the Entabio ZT system. This is a brand new platform class which integrates two key analytical technologies, ICIF, UV, and mass spec, into a top-down solution. We believe this solution has the power to change the game in protein therapeutic development.
There's always a worry that data has changed during analysis.
Imagine being able to determine if a molecule is developable very early in the process.
We need multiple pieces of critical data up front.
Mass information on charged variants would change the game.
Introducing the Entabio ZT system, rewriting the rules of biologics development. For the first time, access multidimensional data not in weeks, not in days, but in minutes. ICIEF separation and UV detection, now coupled with the Xenotop 7600 system. One single run, comprehensive characterization of protein therapeutics across the drug development pipeline. Unstoppable charge variant analysis. New levels of certainty. New speeds of decision-making. Cut out risk. Cut out information gaps. Separate. Characterize. Identify. Win back time for your projects. Win back time for patients. Wherever you play in the pipeline, change the game in modern medicine. Seamless. Unhindered progress. Be unstoppable with the Entabio ZT system from the leaders in the characterization of molecules.
Coupled with the Xenotop 7600, the Entabio ZT system can deliver results in days that used to take weeks to require. A single run, acquired in minutes, can give comprehensive, multi-dimensional data on proteoforms present in protein therapeutic candidates. Coupled with new sophisticated data processing capability within Biologics Explorer software, customers can fully characterize these proteoforms and unlock a new depth of information. I'd now like to introduce Chris Locke, Vice President of Global Research and Development.
New platforms like the Entabio ZT system are a basis for future innovations in technology and workflow evolution. And this is just the beginning. This year, we've seen how continued development of these platforms through new workflow innovation is helping our customers break into new frontiers. For example, our new middle-down protein characterization workflow combines the unique capabilities of EAD fragmentation on the Xenotop 7600 with novel data processing within our Biologics Explorer software. This new workflow can unlock rapid and confident profiling of critical to quality attributes without the need for protein digestion. The result is deeper characterization and significant time savings that can shorten the development timelines and allow for earlier decisions on candidate selection, getting drugs to patients faster. Why is this important? We spoke to one of our customers, Tyler Heibeck, principal scientist from Sutro Biopharma, who told us a story that really brought home the potential impact of these new ways of working.
I don't purposely break rules. I always like to know what the rules are. Not so I can break them, but so I know when and how to break them. Sometimes it's best to follow the rules, but I think it's more important to know when to stick your head up. I will break rules when I think it's productive, when I think it benefits the greater good. Rules exist for a reason, but it's not always a good reason. It could just be because you don't have another answer. I consider myself an analytical biochemist, so I have a broad knowledge of how you use analytical and other physical methods to characterize biomolecules, specifically proteins. I am mainly a mass spectrometrist. Sutro is probably most known for our core technology, cell-free synthesis. There's a rigor that I hope we're known for and I think we're known for. But why are we actually here? We're here for the patients. We're here to make drugs that we hope will save lives.
Sutro has to be fast-paced. We're a mid-cap biotech, right? We're racing other companies that are trying to tackle the same problems that we are, right? And in some cases, it's a sink or swim environment.
I feel like pharma companies usually get a bad rep because they seem like money-hungry sort of entities that don't really have the patient's best interest in mind most of the time. People really do care about innovating cures for degenerative diseases and for cancers, and the people who are doing the lab bench work really do have their hearts in it to help people. I want to be able to say that I contributed to a project that really made a meaningful impact in someone's life. That is the role I see Mass Spec playing out in the future here at Sutro, is helping us achieve that goal.
Where do the unique abilities of our technology give us an advantage? We're all working towards the same goal. We're not competing with each other. We try not to be too siloed. We're making cancer therapeutics. These could go into a person and for an extended period of time, they can live without cancer. Let's say that. Let's say that's the goal. That excites me.
You see the devastating effects of these current treatments, right? The standard of care for a lot of cancers right now is debilitating. So that's Tyler's strength, right? And that he's absolutely making a contribution to humanity.
My uncle, it started out as prostate cancer, and then it turned into colon cancer, and then it metastasized to his liver. And in the end, he decided to refuse care. I do think about him quite a bit. Not that we couldn't cure him, but how much he suffered. There's a 10% chance that a drug I work on will be licensed. And once it's licensed, it may have an impact. Well, I kind of put scientists in two buckets. Experimentalists / empiricists and theorists. I definitely lean on the experimentalist, the empiricist. We're certainly attracted to how we spend our time, how we spend our resources. They're certainly part of our personality. I don't do a lot of stuff half-assed. I do a lot of things ad hoc, but I don't do a lot of things half-assed. So with photography, I dove in deep. I do my best thinking in the morning, and I do it best when there is less distraction. And when I wake up, my mind is more focused. And also when I'm out here, I'm looking at that sharp shadow and contrast and that reflection and how calm that water is, and I'm planning my shot. I'll check my histogram, and I do the same thing when I do a mass spec. You know, the first thing I do is I pull up and I check the TIC, the total ion chromatogram. Getting biopharma done for me is to do some positive purpose in the world, not just exist, to take action, to move something forward, be it knowledge, and if I can, help people, help humanity. Our primary product that we have is called STRO-2. Its targets, and it's an antibody drug conjugate, is the folate R1 receptor. What our main clinical trials focused on is ovarian cancer. The compassionate use for that is something that wasn't even on our radar. We were approached by a researcher that was focused on the very aggressive pediatric leukemia. If your kid has this, you have at most 5 or 7 years with that child, and then they're dead, they're gone.
He identified folate receptor alpha on this small subset of pediatric AML patients. And they treated some of these patients with STRO-2, this folate receptor alpha targeted ADC.
And it works. It tamps down the disease enough that they're now stable for the real cure, which is a bone marrow transplant. Normally they don't do it because the kids are so sick, they probably won't survive the procedure.
Several of my aunts have gone through chemo. I have a cousin that's gone through chemo. When you see these children that are being helped, I mean, it pulls your heartstrings. It truly does.
I was raised by my grandparents, and I lost my grandmother to cancer. She died within 6 months of being diagnosed, and that really sparked something in me. So I got down to small molecule and chemistry. It's exciting to be where I am now and to see how this can impact people in the future.
Because we're really in an exciting point right now. There's all sorts of innovation happening, and I just see that growing. I don't have any paradigm shifts on my radar right now, but that's why they're paradigm shifts. They come out of nowhere.
Just like in biopharma, the landscape of small molecule therapeutics continues to rapidly evolve. As leaders in bioanalytical innovation, our customers look to our technology to lead the development of new quantitative approaches in preclinical and clinical bioanalysis. These are critical steps in getting a new drug FDA approved. And the speed at which this is achieved can have a direct impact on the timescale in which these treatments can subsequently reach patients. Our long-established relationships with customers in bioanalytical labs gives us a unique understanding of the analytical challenges they need to solve to bring new innovative therapies to market. And in many cases, we already have the platforms that can be further harnessed to address these new challenges. One such example is in high-potency APIs, a class of therapeutics that are extremely effective at low doses, and as such need to be quantified in biological matrices at sub-nanogram per mil levels. Now, these protein-degrading therapies have been effective against previously undruggable protein targets in a range of cancers and produce fewer side effects due to their potency at low doses. This year, we coupled the industry-leading sensitivity of our SCIEX 7500 system with the advanced data processing of SCIEX OS software to quantify a class of these compounds, PROTACs, or proteolysis targeting chimeras, in plasma at low picogram per mil levels. Method innovation like this can be immediately implemented in bioanalytical labs to quantify these novel compounds and can inspire the development of any number of new therapeutic strategies in the future. Ongoing development of these new approaches for rapid, robust, and sensitive bioanalysis can ensure that more of these therapies progress through clinical trials to reach the patients who need them. The importance of this became apparent recently when we went to Colorado Springs to visit Illyri Bioanalysis, a high-end bioanalytical CRO. The speed and efficiency by which they operate helps their clients get regulated data to the FDA faster and with greater confidence. Collaborations such as these help us to stay on the forefront of new frontiers.
Things don't always go the way you think they're going to go. You can theorize all you want, but until you actually step into the lab and do the work, you don't know what the outcomes are going to be.
What people don't understand about pharma is how much effort and work it took to get to that point. You usually develop 15,000 drug candidates, and of those 15,000, one makes it to market.
And that's not something that you can just do. Right? It takes time and effort.
We have to get results for our clients. These projects are very important, the studies are very important to them, and if we can't solve the problem, we can't get them the data that they need to continue on with their research.
The balance of efficiency versus running a program that has to be, run to a certain standard. There's so many checks and balances in place.
There's times you have to bend the rules. Being a regulated science company, obviously we don't break the regulatory rules, but when you're developing assays, there are rules to be broken,
We're often pushing the limits of what technology can do. The amount of drug we're looking at is like a drop of ink in an Olympic-sized swimming pool.
You can't stop, that's the thing.
It's the answer to our futures, to life, to better life. Science is so important for our growth and for understanding how everything works.
We're doing the hard work to make sure that the drugs are safe. Once they leave us, they are able to be approved for FDA use.
We're in a contract environment, so we get contracts for all kinds of different, preclinical, clinical.
One day you might be working on a very small molecule antiviral drug and it has, a certain type of property, and then the next day you're working on a, therapeutic peptide, and you have to completely change your approach to working with those compounds. So work is very dynamic.
A lot of the projects that we get in are focused on various bespoke monoclonal antibodies or proteins that are being developed as therapeutics for all kinds of rare diseases. But the similarity of it to a natural protein or peptide in the body is incredibly close. So you need mass spec, and you need to find ways of using mass spec to isolate those individual proteins and distinguish it from something that's already in your body. And that's very challenging.
For any new projects, there's really always 3 stages: method development, method validation, and then there's sample analysis. There's the method development, which is really creating the way that we're going to analyze for the analyte. Whenever you start a problem, you kind of have to have a solution in mind, what you think will work, and you kind of look at the chemical, the compound, what its physical properties are. I think really the first thing is you put pen to paper and you start writing down an approach.
There's always a sense of teamwork when it comes to developing methods. You can bounce ideas off of other chemists and get an idea of, hey, this isn't working, what should I try?
Fix it in pre, that is an ideology I like to think for our method development group. If we get it right during method development, we don't have to fix it in post.
There's often interplay between everyone who's working in the lab to achieve the same goal, which is high quality data to present to regulatory bodies and say, hey, drugs work.
There's so many different things that come with each compound, the size of the compound, what it looks like. Do we think that, we can actually detect it on the mass spec? So a lot of that work goes in up front. You look at binding, different things like that to kind of figure out what's going to be the best extraction procedure.
Once you have a method that's worked through, including working out all the chromatography, figuring out all the masses on the instrument, then you enter a validation phase.
There's regulatory guidances out there, and we need to make sure that we're complying with those, because if we do not, the data could be rejected, which means that a drug could be kept from getting to market, and which means ultimately, somebody may be laying in a hospital waiting for that drug. That could mean they don't get another Christmas or Thanksgiving with their family.
The whole process has to change because it's new technology. You can't analyze these large molecules the same as you do as small molecules. So we all have to think of ways to handle these new issues that we had never seen before.
You want your method to give you real data that you can then use and have confidence that if you were to give this drug to someone you really love, you know what that's doing.
Once that's been completed and we've proven the method works, then we move into sample analysis.
And that's the fun part. That's where the samples come in the back door. There's been a patient in a bed, sample's been pulled from their arm, which is typically, blood that's spun into plasma, and we're gonna analyze those plasma samples. You find out if your assay really works and how well it works. Did the drug get into the body? Did it show an uptake, and did it actually get eliminated over time? And was that reproducible across different patients?
The method's already been validated, so that should just work. And that's the part that really we like, really when things start to kind of just flow.
The excitement is usually when the sample analysis is done and you get to finally report out that complete data set to a client and show them, hey, this is what your drug does.
When the drug comes to market, that's all of your work put together and realized at the end with a real product that's available to patients.
We worked on a Parkinson's drug. We saw footage of patients prior to being on the drug. You could tell they, were not living a good quality life. And then they showed us the same patient after they'd been treated with the drug for a few weeks. And this gentleman had gone from somebody who could barely sit in a chair, jauntily walking from one chair to the other, and looked 20 years younger and just spry again. And I literally brought tears into my eyes. It was like the drug we've worked on, and we've helped these people, get good data for it. We're actually seeing what the outcomes are, and that's huge. You don't always get to see that.
So the work that I'm doing, you can see where it will end up. You can see the impact that it will have on someone's life. And a lot of times, the specific therapeutics that are being developed and that you're analyzing for are more effective with less symptoms and radically changing the lifestyle of regular people every day.
It's those kids who have cancer or, diabetes or something that's affecting their lives. I always keep that in mind because that's why we do what we do every day.
I really hope that in my lifetime, they do come up with a cure, to cancer. Some of these things that have been a challenge for humanity for a long time, we're getting closer. Some of the things that will be solved over the next 25, 50 years, it's going to be amazing.
The biopharma industry has a lot of opportunity for very specific targeting of molecules. There is a lot of development done in cancer research, in very rare disease research that can be targeted with these proteins and these large macromolecules we haven't developed analysis for yet. So from our perspective, finding those new ways of doing things, pushing the envelope for how these drugs can be monitored and determined to be safe is where we lie.
For those in food and environmental science, new guidelines have forced customers to rewrite the rules. The environment around us, the food we eat, the water we drink is all changing. As we continue to understand more about the impact humanity has, we can begin to address challenges that could affect our health and well-being, like PFAS compounds, also known as forever chemicals. Now, PFAS are contaminants of major concern to environmental scientists due to their widespread transport, ability to bioaccumulate, and unknown impact on human health over long-term exposure. In recent years, regulators around the world have started to take notice. In 2022, the EPA proposed new national primary drinking water regulations for 6 PFAS compounds. Now, these interim guidelines advise that drinking water should contain no more than 4 parts per quadrillion of PFAS. Now, this is uncharted territory. These levels of detection are up to 500 times lower than existing methods. Now, once again, working with leaders in this field, we were able to get ahead of these rapid developments and had the platforms in place to take the science forward, meeting the challenge and obtaining these guideline levels. In collaboration with one of these customers, Eurofins, a worldwide leader in testing, inspection, and certification of consumer goods, we were the first to announce a 4 part per quadrillion limit of quantitation on our SCIEX 7500 system. To give you an idea of how small that is, it's like detecting a single cup of water in Lake Michigan. Now, just how big of an impact will this have on science, on commerce, on humans, on you and I? We spoke to Chris Higgins, professor at the Colorado School of Mines, whose lab and research focuses on the fate, transport, and bioaccumulation of these compounds in the environment to find out.
My holistic approach to environmental engineering, environmental chemistry, I think really is rooted in the fact that I was introduced to this topic through the perspective of public health. At the end of the day, what we're trying to do is prevent disease in humans and also organisms. I still introduce myself primarily as an environmental chemist. Why I sometimes like describing myself as an environmental engineer is that it is a little broader in terms of what we cover. Toxicologists usually start at the exposure and the effects, and chemists typically stop at predicting the exposure. But there's some really interesting science that happens during that exposure, how things are being exposed to chemicals, the linkage between the chemistry and the toxicology. As a chemist, if I stopped at just measuring the chemical in the sediment and said, well, it's up to the toxicologist to determine whether that level in the sediment is toxic or not, it doesn't really allow us to get at some of the issues about exposures. The ultimate goal of protecting public health and the environment is why I have that passion to do that. My advisor said, I've heard about these things called perfluorochemicals. I don't really know what they are, but one of the other faculty members here thinks they're really important and they're going to be a big thing. You should plan to study that when you get here. This was summer of 2001. I realized that the sorts of properties they were describing in these papers of these chemicals would be very problematic. And I was like, this is what I want to do. I think what intrigued me most was that they didn't seem to behave the way that other chemicals did in terms of the traditional paradigms for organic contaminants. Broadly speaking, we've talked about organic compounds that are small, they travel quickly in groundwater, but don't really bioaccumulate. On the flip side, we talk about very large organic compounds like PCBs and DDT that don't move, they stay in one place, and they do bioaccumulate. What was really intriguing to me in those first few years was that there are these compounds that do apparently move relatively quickly, and yet they bioaccumulate. In those early days, when we were first doing some of these first measurements, we were using what we now call targeted LCMS, where we have an authentic standard of a compound. If we didn't know that something was out there, and we didn't have an exact identity, it was very difficult to measure something that you don't know what it is. That's been a fairly recent development in the PFAS community, is recognizing that we can go look for things that we don't a priori know what they are. This broader interest in why we care about these chemicals really took off in 2016, when there was this recognition that there's a lot of communities out there that have these chemicals in their water. There were two moments that I very distinctly remember realizing this was going to be a much bigger problem. The first was when some of the UCMR data started coming out. If I were to look for foam impacted sites, I would be looking for PFHXS. So I pulled up the UCMR list and looked for foam impacted sites. And lo and behold, I found my hometown. And I realized if there was contamination of foam at the tiny little airport near the town that I grew up in, and it was enough to cause the groundwater to be contaminated, to cause the drinking water to be contaminated above the levels of reporting for the UCMR, we're in for a whole world of hurt, in that this was a much, much bigger issue. There are many, many more sites that are going to be out there. The second, when I realized that there might be broader interest, was when I got a call from a BuzzFeed News reporter when the final health advisories came out. The real story came up when I started looking at these firefighting foams and recognized that if you look beyond PFOS, everything else that was intentionally added to the firefighting foams were precursors. We're talking about compounds that are not fully fluorinated, but have the potential to transform to the fully fluorinated perfluoroalkyl acids, which is what we are mainly focused on nowadays for toxicological assessments. These polyfluorinated substances we often refer to as precursors because they will form, under the right conditions, these perfluoroalkyl acids. And that can be in the environment, it can be in the atmosphere, it can be in water, it can be in soils, it can be in our bodies where those transformations happen. But those transformations can and do happen. That's why we care about these precursors. It's somewhat surprising that we're still trying to answer some of the same questions that I think we started out with, which is that there's a lot of these compounds out there. How do we measure them? How do we estimate them? How do we identify them? And I do think we're going to be working on this for a long time to come.
The reason I became an environmental chemist is I want to do things to make, as corny as it sounds, the world a better place. In order to do that, you have to recognize what the real problems are. For me, it's doing the science that's more broadly applicable. It's the things that the communities down in Colorado Springs or whatever are struggling with, and we need to try and help them. We are a society that uses chemicals for everything. We're not yet at the point where we understand how chemicals interact with our bodies or with other biological systems or the environment. One of the pieces of advice given to me is whatever you do, make sure you study something that matters.
It's paying attention to the world around you and what are the needs. And I'm sure there'll be other needs when the PFAS story is over, if it ever is over.
I think as you've seen, we don't do things incrementally. When we innovate, we step change. We rewrite the rules, we break into new frontiers. Step change means new platforms, delivering transformative innovation like the Intalbio ZT system, which we're very excited about. Platforms that in our customers' hands can have a tangible impact on people's lives, just as we've seen today. It's the beginning of a journey where we'll build upon the core capabilities of our platforms that you've seen today through the creation of new workflows, new approaches, and new partnerships. Today, it's just the beginning of new disruptive approaches to biotherapeutic characterization based on the Intalbio ZT and Xenotolf 7600 platforms. As we continue to innovate on both our ICIEF and accurate mass technologies, we can envisage entirely new classes of workflows that can combine our widely trusted CE technology together with next-generation mass spec approaches and further evolve the landscape of fast, safe, and innovative biotherapeutic development. And as we think about the SCIEX portfolio as a whole, we will continue to strive for hardware, software, and ultimately whole workflow innovation that can unlock new markets and opportunities for our technology. For example, just last month, we opened the new EchoMS Center of Excellence at our headquarters in Framingham. We collaborated with our friends at Beckman and Gene Data to bring industry-leading automation and data processing together with EchoMS. We created an end-to-end high-throughput screening solution through this collaborative venture. And yes, the world is changing. And we at SCIEX look forward to this new frontier. We'll be there alongside our customers just like today, and our technology platforms will be there in their labs as they bring new therapeutics to market, ensure the safety of our food and our environment, cure rare diseases, and what I expect will be more outstanding scientific achievements in the years to come. We're in this together. Let's rewrite the rules together, pursue new paths to the next scientific frontier. Everyone, thank you for coming today. Thank you for your time. We're going to set up on stage for some questions. Thank you.